Hey kids, on clean off my camera. It's been a slow Sunday, Corbin. Pretty slow. Catching up on a whole bunch of stuff. Look at that stack of papers. That's just one stack. Yeah. All right, kid. Let's get down to it so we can make the most of this hour. Come on. Alrighty. All right, let's go ahead and start while I'm waiting for questions. Go ahead and make the little charts. If y'all haven't made the chart, that's probably going to be one of the best things you can do for yourself right now. Patriots better. That's the only thing happening today. Don't watch football, so couldn't really comment on that, Max. Sorry. I don't watch really any kind of sports. I watch Formula One with my husband. That's it. Hey, William. All right. I'm not ignoring y'all. I'm writing. So just give me uno memento or minuto or minuto or whatever it is in Spanish. Just uno minuto. I don't know what it is. All right, let's see. Glad you're doing this, Miss Good, because I didn't really get this unit as much as photosynthesis. Oh, no. Okay. All right. Hi, don't kick the cactus. Hey, Michaela. Okay. All right. All right. So cell respiration. Let's go ahead and just break it down in a couple of steps. Don't forget your note card cheat sheet you get to have. The small note card, little index card, front side only. So only one side. I don't care which side you use. Some are like, ooh, can I do it on the lined side? I'm like, I don't care. A side is a side is a side. Put it on the note card, handwritten only. You do turn it in with your test. Put whatever you want on it. I don't care. Okay. All right. Just as long as it's handwritten. I feel like I need to specify handwritten directly on it. Not like handwritten and then Xerox copied to make it smaller and then taped on it. And the only reason I'm specifying that is because I actually had a student do that once. Yeah. They Xerox copied the test review and shrunk it and then fit it on their note card. And then they brought a magnifying glass. Yeah. It was crazy. Never mind. I can't hear you. Okay. All right. So cell respiration. Let's go ahead and break it down just into it. It was smart. I did let them use it, but then I started clarifying after that child. So yeah. I also had a kid write in 2 different inks and bring the filters. So the red filter and the blue filter. So they like had a double layer going on, but they just had to put the filter over it and they could see it. Yeah. I let that kid use that time too. Some pretty smart kids. All right. Cell respiration. It has to start with glycolysis. You're not late. It's fine. Have to start so we can use a note card, but yes, only one side, sweetie. Handwritten only. Cell respiration starts with glycolysis in the cytoplasm where it takes in glucose and converts it to pyruvate. You get ATP and NADH. You got to keep in mind glycolysis is anaerobic. All right. So from glycolysis in the cytoplasm, you run into the fork in the road. And at that point, this is determined based on how much oxygen do you have supplied to your cell right now. Do you have a pretty big stock going, or are you about to require too much ATP that you're going to run out of your stock too fast? Could we fold up our paper and then glue it? No, but good try. Yeah, good try. I had a student once take their index card, steam it somehow, and separate it out into layers. So they separated the layers of the paper. And so they got one note card, but it ended up being almost a whole piece of paper because they steamed it and separated. And they're like, you just said one note card. I'm like, are you freaking kidding me? Who sits there and thinks of these different options? So when they ask for the products of glycolysis, we just say the net gained ATP or all the 4 ATP molecules. That would be specific. Like I would specifically ask you, how many ATP molecules are net gained at the end of glycolysis? Or I would say, how many ATP molecules are made in total or gross? Like I would be very specific in the wording if I asked you, okay? So anaerobic happens in the cytoplasm. You get to your fork in the road. It's going to happen anaerobically if you do not have enough oxygen in storage. So if it's anaerobic, that's when you're going to enter into fermentation. Okay. And fermentation itself also has 2 steps. Fermentation is going to stay in the cytoplasm. All right. Try and write that in here. All right. Because you only get to exit the cytoplasm if you have oxygen available. So if you don't even have oxygen, you're stuck there. All right. So fermentation, you have 2 options. You taking your clothes upstairs? Are you going to bed?
Good night.
Good night.
Did you get a new jacket?
No, I just got cold. Say hello.
Hi.
She just magically needed to come in here. Not because she wanted to try and get on the live stream.
No, because I need to get my clothes.
Good night.
Good night.
Did you finish packing your lunch?
Yes.
Okay. William says good night.
Okay.
She said, okay. All right. So here's the thing. In fermentation, so if you do not have enough oxygen storage, remember fermentation is going to be short term. Doesn't last very long, but you always kind of get in it for first few minutes of working out, no matter what. You can't avoid it. So lactic acid fermentation, and the ultimate output of lactic acid is just that it converts 3 carbons to 3 carbons. Like it literally moves the carbons around, moves the oxygens and hydrogens around, and it just changes pyruvate into a different molecule. So that's all it's done. Specifically, this is only going to happen in animals. All right. So you've gone from glycolysis to fermentation if you don't have enough oxygen. This one only happens in animal muscles. That's literally the burn you feel in your legs when you've worked out. It is literally burning because you have acid buildup. Okay. Stretch before, stretch after. Very short term. Okay. If you are, let me go ahead and write this. This is getting a little sloppy, sorry. If you are bacteria or fungi. If you're a bacteria or fungus as in yeast for your bread, then you're going to go through alcoholic, which releases alcohol and CO2. Again, remember this because this is what allows bread to rise, right? The CO2, as it sits and they start to ferment, the CO2 gas actually starts to push the bread up. And then also that's that smell that you're smelling. It's actually fermenting alcohol. But the second you go to bake it, the alcohol evaporates off, the CO2 releases, and you're just left with its holes. Okay. right, so those are the only two types of fermentation. Lactic acid if it's animal muscle, alcoholic and CO2 released if you're a bacteria or a fungus. Should we consider glycolysis part of the process or a separate process? Yes, glycolysis is there no matter what. You can't not go through glycolysis. It's the original process. And here's the reason fermentation happens. Because it has to recycle. Let me go ahead and just write that in here. It recycles the NAD. Okay. It recycles NAD. So NAD can go back up to glycolysis and get made. So then it can go back through fermentation. The reason it recycles the NAD so that this process can keep going for those first few minutes of working out, and you still have enough energy, it's not a lot, but you still have enough energy to breathe, to move your legs, to run, okay, for your heart to beat, for your nerves to transport, stuff like that. Does lactic acid fermentation release anything? Lactic acid. Like that's what it's releasing. Isn't there more fermentations, but these are the only ones? Yeah, there's hundreds of paths. We only talk about 2. So the anaerobic processes would be glycolysis and fermentation. Yes, those are the only 2 anaerobic. Those are the only 2 that happen in the cytoplasm. After that, if you have oxygen available, like you do right now, for the necessary things you're doing right now, called long-term energy, that's where you're going into your actual aerobic respiration. So the aerobic, okay. Okay, your aerobic respiration. I kind of wrote this twice, but after glycolysis, your long-term aerobic respiration is going to take you into the mitochondria. You're going to go directly into the Krebs first, which happens in the matrix. Okay. Krebs happens in the matrix. That's your first step. The Krebs cycle, just so you know, releases CO2 and energy. Okay. The purpose of the Krebs is, please slow down, too much to process at once. I don't even know who you are, Creek. Who are you? Because it just says Creek. Hello. Oh, it's Gage. Okay. Oh, Gage, keep up. You're fine. Okay. All right. Krebs cycle. Remember the Krebs cycle? It's the cycle that it happens in the open space matrix. And we'll go ahead and compare chloroplast and mitochondria in just a second too. So here's the thing. The Krebs cycle takes what was just made. Both of these processes take what was made, pyruvate. Over here, the pyruvate comes in. It's immediately going to be converted. CO2 gets released and energy. Think about all the energy brothers and sisters that belong to that family. ATP, NADH, FADH2. Those are all brothers and sisters that belong to energy, with ATP leading the path. From there, gas is always released. Gas doesn't get pushed to the next step, but whatever was made in the step before, guys, gets used in the next step. Pyruvate got used. Now our energy is going to get used. So now our energy is going to go into the final step called the ETC, electron transport. So that happens in the cristae. During cell respiration, is fermentation constantly happening, or does it only happen separately? Fermentation only happens if your body needs it to happen because you needed fast energy supply. Like right now, you're doing some push. Give me some push-ups. Now there, come on, right? No, it's only if your body needs it. All right, it's always going to just sit there and go through cell respiration. Miss Good, is the starting molecule of the Krebs the acetyl-CoA? Acetyl-CoA, good job, Maria. Cell respiration is aerobic. It is not anaerobic. Oh, you're asking me a question. Sorry, Nanya. Cell respiration is aerobic. It is not anaerobic. Cell respiration, we talk about it having an aerobic and anaerobic stage, like the whole thing. Fermentation is anaerobic, so if there's no oxygen, then it happens. Yes, they're all kind of answering your questions for you. All right, so let's kind of start again. Pyruvate's made. Long-term energy goes into the mitochondria, goes into the Krebs cycle. Pyruvate does. Acetyl-CoA, sorry, let's get very specific. CO2 gets released. That's waste. Energy gets made. That energy goes into the ETC, which happens in the cristae. And it uses, I'll get to your question, Nicholas. That ETC uses up that energy, rips off the electrons, we'll go through that, makes H2O and ATP. Those are the ultimate jobs of ETC. Make water, make ATP. Make water, make ATP. Is all fermentation short, or is one of the two long-term? They're all short-term. Fermentation is short-term. Okay, so general little chart, a little bit more detailed than I had on the board, but it just says where it happens, what it starts with, what it makes, if it's aerobic or anaerobic, right? And what are the products? Remember, the products from one step are what you are used in the next step. Pyruvate, acetyl, energy, etc. Okay, hold on. Acetyl-CoA. Let me go ahead and draw that. What's the difference between cristae and intermembrane? Nothing's the difference. Cristae is just the name I want you to know it by. That's its true name, is cristae. Do we need to know about the acetyl-CoA? What is it? There is no difference, I guess. Yeah, no difference, Nanya. Ooh, that definitely leaked through. Okay. What am I going to do? Okay, so glucose. 6-carbon molecule. I will in a second, Corbin. All right, 6-carbon molecule goes, bam, let's break it. Glycolysis, breaking of glucose. Then you're left with 2 3-carbon molecules. Pyruvate. Okay. Your 3-carbon molecules of pyruvate, if they're going into aerobic, okay, if they go into aerobic respiration, at that point, what you actually enter before they even get thrown into the Krebs cycle. So you've gone from glycolysis, you have oxygen, so you get to go into the mitochondria. And at that point, before you actually get to go into the matrix and start through the Krebs cycle, the pyruvate gets attacked. Ooh. And immediately loses a carbon as CO2. But the reason it loses a carbon is because an enzyme comes up and rips it off. That enzyme is coenzyme A. So coenzyme A comes up, rips that off, and takes its place. And you're left with 2 carbons in this CoA. We call that acetyl-CoA. This is the molecule that begins the Krebs cycle. So that goes into the Krebs, also known as citric acid, because that's the first thing it's made. Okay. So that gets thrown. As it goes through the Krebs cycle, it releases those other 2 carbons of CO2, releases energy, and energy. Okay, that's the stream lag out, or is it just me? My side is doing just fine. Like, it doesn't show lag on my side, but sorry. Okay, so here's your Krebs as in detail as you need to know. Okay, Corbin? Glycolysis, broke it. What do we get? Pyruvate, 3 carbons. This enters the mitochondria. Let's go ahead and do that. Okay, do. Let me go ahead and matrix. Ha! This entered the matrix. From there, your pyruvate lost to CO2. Why? Because this bully, this enzyme, ripped it off and took its place, leaving you with acetyl-CoA. That got thrown into the Krebs cycle, and as part of the Krebs cycle, released it as CO2. See, those 2 carbons got released as CO2. Do you see that? So what gets recycled ultimately? The coenzyme A. Why? Because he's going to come back up here, grab another pyruvate, rip off the 1 carbon, take his place, and go again. CoA keeps getting reproduced, keeps getting recycled. As a release of CO2 happens, energy. That's what all those are. NADH, energy. ATP, big energy. FADH2, little brother energy, right? That's what just got made. And now our FADH2 and NADH, they are going to go from the matrix into the inner membrane. Okay. I don't like that drawing. I'm gonna take them and go to the other side of my page because I can't really see that very well. Oh. Okay, so let's. Okay, so I'm not going to get into the whole detail of drawing a phospholipid bilayer, but this would be the outer phospholipid bilayer. So, yeah, you'd have phospholipids. You'd have another phospholipid bilayer right here, and that would be our inner membrane, or what y'all are supposed to know it as, cristae. Okay. All right. So you've got outer, some space between, cristae, and then the open space down here would be the matrix. So in the matrix are NADH and our FADH2 were just made. In the matrix, this was the Krebs cycle. This just occurred here. So we're going to take our energy. Okay, we're going to take our energy and we're going to throw it into anaerobic respiration is glycolysis and fermentation. All right, so let's look at the ETC because that's our last and final step. So the first thing is energy is going to be broken. Okay, so we're going to take NADH and rip off a hydrogen. We're going to take FADH2 and rip off the hydrogens. Every time we rip off hydrogens, we also rip off electrons. So these electrons now travel. Okay, so energy was just made in our last step. Energy gets used in the proteins of our ETC. Rip off hydrogen, rip off hydrogens, transfer electrons. That's why it's called the ETC. Then those electrons are thrown back out. And now those 2 electrons get added to oxygen, hydrogen, and make water. Okay. So general question. What's the purpose of the electrons in the ETC? To make water. Okay. Now, when we stripped off the hydrogens, yes, we released electrons, but we also released hydrogens. Right? Those hydrogens started flowing. See where they're going? They're going in that space between the two membranes. That space has a name. It's called the intermembrane space. Inter, I-N-T-E-R, means between. So the between space, between the membranes, okay? So the hydrogens start flowing. They start flowing. They start flowing. That's what... My dog is whining. They start flowing. That's a whole lot of hydrogens in a very small, cramped space. They don't like it, so they want to find the revolving door to get out. That didn't change from photosynthesis. The revolving door is your ATP synthase. Hydrogens flowed through, allowed for ATP to be made. Okay? That's it. ETC, break energy, release electrons, make water, hydrogens move, make ATP. Just the ETC makes on average 32 ATP by itself. On average, for every 1 glucose molecule, it can make on average 32 by itself. From glycolysis and from Krebs, you get on average approximately 38 ATP from 1 glucose molecule. All right. So Colin, again, so Corbin, those were the two, like that's as detailed as you have to get on Krebs and ETC. And glycolysis, that first part was as detailed as you needed to get. Glucose breaks into 2-pyruvate, gets you 2 net ATP and NADH. And anaerobic, Colin, is just glycolysis is anaerobic and fermentation is anaerobic. How many molecules of ATP produced from a single molecule? Just answered that. Okay. So on average, 36 to 38. That is just an average number. Is long-term energy an aerobic process? Yes, that's what you're doing right now. Your long-term is considered what is necessary to just maintain life. And you have to do that aerobically, or else you're sitting there burning, creating a lot of lactic acid and hurting yourself. I think 4. So, Maria, where are you saying I think 4 to, as in the ATP? It's 36 to 38. So the electrons are ripped off NADH and FADH2. Yes. Because when you take a hydrogen, hydrogens are bonded to the other rest of the molecule with electrons. So when you rip off the hydrogens, you ripped off the electrons too. Does the system also use the fat long-term energy? It can. That's why people who fast are still breathing. They're still thinking, they're still walking. Because even though you're fasting, you're not bringing in glucose, right? You're not bringing glucose into your body right now, right? It's still going to go to your liver for glycogen, and it will attack your fat cells. And there are different pathways in your body that actually can take fat and protein, flip it all around, and get it into your cell respiration process too. So it doesn't have to start with glucose. What? Mind trip. Don't think about it. All right. So 32 ATP from ETC process. Yes. 32 from just the ETC. 36 to 38 overall. How does hydrogen aid in the production of ATP? It's the same thing as photosynthesis. Hydrogen starts to build up. It doesn't like it. So it has to find the revolving door of the ATP synthase. So it activates the enzyme. That enzyme then makes ATP. And notice the hydrogens don't get, remember, hydrogens don't get into ATP. That's just phosphate. The hydrogens came through and they got created into water. 2 net gain from glycolysis, 4 ATP total. Or is that a question? Yeah, 4 gross ATP, meaning the max. But you only get to keep 2 because it took 2 to break glucose in the very beginning. So you have to like pay your body back. Is there a difference between cell respiration in plant and animals? Nope. Cell respiration is the same in both. Okay. Whenever we draw our plant cells, mitochondria are in there too. They go through cell respiration just like we do. But once they have their first leaves, photosynthesis takes over. So while a seed is in the ground, cell respiration, just like we do. They break down sugar. Sugar's stored in their seed. Why do you think when y'all eat peanuts and stuff, y'all are eating the seeds? It has a lot of carbs because that's all sugar. Because the plant was actually breaking that sugar down to survive until it produced its first leaf and could photosynthesize. Anna, Zach's right. Plants photosynthesize and go through cell respiration. We actually get in more detail in that second semester whenever we talk about plants separately. For fermentation, Nicholas, I don't know I'm not sure what you're saying. Are you saying fermentation only gets you 4 ATP? Fermentation only gets you 2 because it's the same 2 that you got from glycolysis. Okay, fermentation only gives you 2 ATP because there is no ATP actually produced in the fermentation step. You only got it from the glycolysis step. Wait, do we not have prefix suffix? Not on this one. The prefix suffix was photosynthesis. All right, Nicholas, you're going to have to catch me up. I hope I answered your questions because yours is so separated, it's hard to follow. What is the part where the pyruvate is broken down to a 4-carbon molecule and then recycled? Krebs. Pyruvate was never a 4-carbon. Pyruvate is a 3-carbon. What you're thinking of, if you're looking at the test review, the Krebs cycle, the pyruvate was added to a 4-carbon, but we're not going that in depth. The pyruvate was added to a 4-carbon, and that 4-carbon got recycled. But yes, those recycled parts happen in the Krebs. Do we need to know the specific steps of the Krebs cycle? What you just saw and what is on your drawing are as specific as it gets. The acetyl-CoA goes into the Krebs, gets released to CO2, and it produces energy. Okay, Nicholas, you good? Okay. All right, if we compare a chloroplast to a mitochondria, I'm just going to kind of do this real quick. Just be able to see the difference. All right, chloroplast, thylakoids, right? Thylakoid stroma, mitochondria, matrix, cristae. So the thylakoid, which is the membrane, is like the cristae, which is the membrane. Those are equal to each other. The open space, which is the stroma, is like the matrix, which is the open space. So those are equal. The stroma is equal to the matrix. The thylakoid is equal to the cristae, if you're comparing the two. Because the cristae and the thylakoid are membranes where the ETC is. The stroma and matrix are open spaces, so a cycle can happen there. Will there be 20, 25 questions? You can always anticipate up to 30 questions for a test. Do we need to compare photosynthesis and cell respiration? Something like that, perhaps. You might get a question like, where do they take place? The matrix is like the blank, right? Or the Krebs cycle is like the Calvin cycle, something like that. It'd be a very general question if I asked it. Okay, Maria? As far as drawing, that's it. Like, really, that was it. If you know your steps, glycolysis, what is it? Break sugar into what? Pyruvate. Get you how much ATP? Net of 2. Where does it happen? Cytoplasm. Does it use oxygen? No. What if I don't have oxygen? Uh-oh, you got to stay in the cytoplasm and go through fermentation. But I'm an animal. What do? Make lactic acid. Why? Because it burns. What if I'm a bacteria? What if I'm a yeast? You're going to make alcohol and CO2. You're going to help us with bread and alcohol. Why do we do these things? To recycle energy. Back here. Glycolysis breaks glucose into what? 2-pyruvate and a net gain of 2 ATP. It's anaerobic, happens in the cytoplasm. But I'm just living my daily life. What am I doing? Aerobic respiration. What is that? It happens in the mitochondria. It goes through the Krebs cycle. What's the Krebs cycle? Happens in the matrix. What does it do? It takes pyruvate, makes acetyl-CoA, releases CO2 gas and makes energy. Why does it make energy? So it can go to the cristae, into the ETC. Why does it go there? Make water and ATP. Yay! Okay. You probably are going to overthink it, Max. Yeah. Do we need to compare? Okay. Can you once do the chart like how you did? Okay. I that was the very first thing I did in Anya. Were you not on here? And that's what I just showed to you again. Okay. So just freeze the screen afterwards or something like that. All right. So if you got the chart, right? And then for those that needed more of the like specific kind of picture, right? Glucose, 6 carbon. Cha! Why did we do that? Or how did we do that? By using 2 ATP. Oh no, we had to use energy. Energy did what? It broke it. What do we get? Pyruvate. What do we do with it? Make it into acetyl-CoA. Here's the Krebs cycle. Ding. Ding. What am I doing in the Krebs cycle? I'm recycling this guy. Why? So he can keep bringing in pyruvate. Why? Because the Krebs cycle makes CO2 and energy. What does the Krebs cycle do? Make CO2 and energy. Why does it do that? Because the energy goes to the ETC. Yes, Brett. What's that ETC you talk about, man? There you go. Energy. Release electrons, break off hydrogens. Release electrons, break off hydrogens. Hydrogens hate being pushed. They go through the door. What do electrons do? Help make water. Where does it happen? The cristae. That be it. I mean, really, it really is. That's it. Like the ultimate question you have to ask yourself, especially since you get a cheat sheet, people. I mean, come on, is do you have this kind of chart on your cheat sheet? Yes or no? If you don't, I can't save you. Like, there's nothing else I can do. I can pray for you, but I can't save you any more than that. Okay? Do you have this chart on your note card? If you do, you're doing great. Do you have more space because you write small? Awesome. Then maybe you want to draw it out. I would probably think the more specific drawing I would attempt to do would be the ETC one. And then maybe if I needed to not draw it, but just write it out, that the thylakoid equals the cristae and the stroma equals the matrix. And that's it. So each energy molecule goes to a separate protein in the cristae. Yes, it does. I'm not going to bother with being that specific. But yes, it does. It's just both are being used. You don't, at this level, you don't need to worry about that they're going to different proteins and what are those proteins' names, and you don't have to worry about that. So. Yep, that Mr. Pibbs sat out way too long. It is a little bit flat. Yeah. What happens to 2 electrons in the ETC? Bam. Let's follow it. Right here. They got ripped off, right? Here's your 2E. I know that doesn't really look like an E, but I promise it is. Okay. Follow your 2 electrons. So it goes through the proteins because that's why it's called the ETC. They transport them. They get spit back out. They find an oxygen. They find hydrogens that just flowed through. They combine, hold hands, and they make water. Electrons are given to oxygen, and oxygen's like, where are my hydrogens? Where are my hydrogens? And as soon as the hydrogens come through the door, it grabs them and is like, ha ha ha, and says, I made water. Flat soda is nasty. Oh, it's tasty? No. Ooh. Is the cristae in the matrix, both in the mitochondria? Yes. Murphy's. Savannah, is that you as in Savannah Peters? Because it says Savannah Murphy. Yeah. Yes, just like you had to know the thylakoid and stromal for the chloroplast, you have to know the cristae and matrix for the mitochondria. Is the ETC or Krebs more important to draw on the cheat card? I would say the ETC, but I would draw it very, very simply, like just simple, because the chart, guys, for the Krebs, takes acetyl-CoA, makes energy and releases CO2. It's basically what you already have on your chart. You might just include that it's acetyl-CoA instead of pyruvate. But yeah, I would draw the ETC. If you're going to draw anything, the chart might just be enough. I'm just sitting here waiting for questions at this point because seriously, guys, there's nothing else for me to cover. That was it. Yep. I don't know about y'all, but our fence blew over because the rain. That was fun. Part of our patio furniture too. Any other questions I can answer? I posted quizzes, Quizlet, Bozeman video. I'll post the live stream, all of that on Classroom. The other stuff's already there. So you have that. What days are band gone? Tuesday through Thursday. The smaller one, like a 3 by 4, 3.5 by 4 or something. Normal size. Oof, that sucks. It does suck. So band, you're missing Tuesday, which is a lab you'll make up. Wednesday is beginning of DNA notes. Thursday is just continuation. So band, you're essentially missing a chunk of DNA because DNA is only 3 days of notes and we're done. Yeah. But it's okay. Band, just breathe. This happens every other year. Y'all survive it. Okay. I think you already mentioned this, but for long-term energy, what will your body use? Your body uses glucose. That's what cell respiration is, uses glucose. For a short-term energy, well, let's be specific. Short-term energy molecules, short-term energy process. The process is called fermentation. But your body, if it needs just short, quick term, it's going to use the molecule glucose to throw it in the long-term process, respiration. And then your body will store fat as a long-term solution as a molecule. Okay. So when you rip off the hydrogens off the NADH, you also, yes, electrons, NAD, hydrogen, held together by what? The electrons. When I rip off the hydrogens, I ripped off the electrons too. The electrons go through the ETC, the hydrogens float into that intramembrane space and get stuck and find the revolving door. Oh, you're oofing about the fence. What day will our last test be? Last test? You mean our next test? Like DNA? It's going to be literally the Friday before we leave. I normally like to do it on Thursday, but I won't be there for part of the day Thursday because I'm in a training. So it'll be that Friday before we leave for Thanksgiving. And if y'all are leaving early, sucks for you. You have to take it when you come back, which sucks. We can talk about that later. Can you please hold up the ETC drawing again? Outer membrane, space, cristae, energy, rip off electrons and hydrogens, create water, create ATP. So aerobic in the mitochondria is long-term. Yes. Yes, it is. I'm going to get my last name officially changed tomorrow. I don't know what you mean, last name officially changed. Are you not going to be Peters anymore? Aerobic is long-term and anaerobic is short-term, yes. Yes. Anaerobic, only a few minutes. Why does the synthase protein look like a flask? It's just because that's the best I can really draw it. I mean, if you want me to get all kinds of like technical. You want me to get kind of technical, even though I really can't draw it at all? That's actually your ATP synthase. You got a top part that moves and rotates, a passageway tunnel for hydrogen. This acts like a gear shaft. As that turns, it starts this turning, and then that little piece helps make ATP. So it's like, so just in case you wanted to know. And all those pieces actually have names. Yeah. The stratter, the rod, the knob, yeah, the rotor, yeah. They all have very specific names, and you have to know them if you're in UIL. Yeah, isn't that fun? All right. Corbin, speaking of which, you need to let me know if you're coming Saturday. Ananya, same thing. Alex, if you're on here, same thing. For UIL this Saturday, we're in Hazel. Looks like a crank mix with the Gatling gun. Yep, pretty much. Pretty much. All right, any other questions I can get for you guys? Because that's all I'm doing at this point, is just answering y'all's questions, because there's nothing else I can draw or really say. Sorry, I'm realizing I need to send an email to someone. We have the lab report due the 16th. Yes, you actually get a work day in class on November 15th. Since I'm not going to be there for part of the day because of another training. I go to so many trainings lately, I swear. Yeah, y'all actually get a work day. You know, the rubric is in the classroom. Good and bad lab report examples are in, sorry, not classroom, but your drive. And it's due November 16th at 11:59 p.m. So, and y'all are free to come in, ask me questions, look at it, that kind of thing. I'll probably just end up making a little video and posting it on Classroom as far as how to make a graph with y'all's data. Okay. Haven't started, better do it. As far as if you sit down and try and do it all at once, you'll probably sit down for 3 to 4 hours. If you, or you can spread that out over time. But if you're going to sit down and just do it, just like, well, that 3 to 4 hours depends on how quick of a typer you are. If If you're a halfway decent typer, 3 to 4 hours. If you type really, really slow, it might take you more like 4 to 6. So, okay. Okay. Are we done? Is there always O2 in the matrix? Yes, because the water is constantly being broken back down and creating oxygen. Products of alcoholic fermentation are alcohol and CO2. Yes. Yep, that's why bread rises. Why don't I see the good, bad lab reports? I don't know. They're just in drive. Yes, it's very specifically ethyl alcohol. You can just say alcohol. Y'all don't know the different types of alcohol. Just say alcohol. Why would anyone come rate a lab report? Come rate a lab report? It is the spawn of Satan. Rate. Why would anyone create a lab report? You have to. It's your lab assessment. I couldn't find the good. How are y'all not finding them? I'm pulling up drive, so just chill. Chill, children. It's very quiet. Be very, very quiet. We's hunting rabbits. My drive's being really slow coming up, so hold on. It should be in there. If it's not, I guess I'll post it right now. They should have already been in there like a long time ago. Wow. This is taking forever to come up. Yeah. All right. That's awesome. Oh my goodness. I think it's like, no, you're doing a live stream right now. We don't really think you need to go very fast at all. Yes, the lag is happening. Okay, I thought they were before entering. Yes, they're just in the, yeah, they're just in the folder. Last test grades will come back tomorrow. Last test grade you'll see tomorrow. Krebs. Pyruvate turns into acetyl-CoA. Makes CO2 and energy. Krebs, aka citric acid cycle. Why? Because the first product it actually makes right here in this little spot that has nothing, because I don't need you to know how specific it is, people, is citric acid. If you want to know, sure, you can come in and I'll show it all to you. I'll write out all those molecules. I'll show it all to you. I'll show you the little 4-carbon molecule called oxaloacetate that it actually gets attached to. Then when it makes it and then it gets broken back. Yeah, we can go through all that if you really wanted to. You don't need to know it. Hint. His isn't a hint. Okay. I don't know what you mean. This isn't a hint. Oh, okay, good. This is far out, but after Thanksgiving break, what are we doing the Monday week back? Hint, I won't be there that day. We start something called protein synthesis. You want to... Max, you're going to have some afternoons, that's for sure. Whenever we come back, so we have cell respiration, then we do DNA. Then, because realize it, guys, we have 2 weeks, and then we go for Thanksgiving. Freak, yeah. So we do DNA. Work on your lab report test. When we come back, we do protein synthesis test. Then we do cell cycle test. Yeah, you have 3 more tests in my class. You've gotten a lot of tests this quarter. So, all right, are we good? And, oh, yeah, fun. I remember you telling me that. All right, guys, are we good? Since I'm missing several days, what do I need to make up again? Y'all will have to make up a lab. Nothing for you to write. It's just y'all have to come in during Omega and take care of that. And your notes, I will give y'all's notes tomorrow in class, Nicholas, after the test. So y'all just need to look at notes. That's it. There's no note quiz for this chapter. Thanks, Miss Good. Yep. Peace.